Namaskar. Today we are going to discuss about the difference between polyclonal antibody and monoclonal antibody. We have already studied the polyclonal antibody and the monoclonal antibody in our earlier classes. Today we will discuss their difference, the difference between them point by point. As we know, the antibodies are produced by the B lymphocytes, the plasma B cells. They produce antibodies. So when these antibodies are produced by multiple clones of B lymphocytes, then we call it as polyclonal. And when it is single clone of B lymphocytes, we call it as monoclonal. So you can see the difference in the picture itself. You can see here the single color, and here you can see multiple colors. So multiple clones produce polyclonal antibodies, single clone produce monoclonal antibodies. So you can see, if we discuss about the production, for production of polyclonal antibody, the method is simple. We have to inject the antigen into the animal. Then that antigen will activate the B cells in the animal. We then have to collect the antibodies from the B cells, from their blood, and those antibodies are polyclonal antibodies. Whereas in case of monoclonal antibody, we will inject the antigen to the animal, we will collect the spleen cells from which we will harvest the plasma B cells, and we will also take the myeloma cells and we will hybridize the plasma B cells with the myeloma cells, and we will get number of hybrid cells, and we will allow them to proliferate, and we will get a number of hybrid cells, and we will screen them for the desired antibody. And suppose this hybrid cell consisting of the plasma B cell and the myeloma cell acts against the specific epitope in the antigen. So we will proliferate this hybridoma. So this hybridoma will be grown to produce large batches of antibodies, and those antibodies are monoclonal antibodies. So this technique is called hybridoma technique. So based on this, we can differentiate the monoclonal and polyclonal antibodies. So it is simple that polyclonal antibodies we can prepare by just immunization of the animals. So you can find them in the serum in response to the antigen, whereas we have to use the hybridoma technology for production of monoclonal antibodies. So the technology is relatively easy in case of polyclonal antibody and highly complex in case of monoclonal antibody. Similarly, the skills, fewer skills are required in case of polyclonal antibody, and higher level of skills are required for monoclonal antibody. And when we produce, the scale is limited for polyclonal antibodies and scale is very large in case of monoclonal antibodies. Accordingly, the cost factor will come. So polyclonal antibodies, they are cost effective, and monoclonal antibodies are expensive. Production time is short in case of polyclonal antibody, 2 to 3 months only, and more than 6 months is required. So production time is long in case of monoclonal antibody. Since the production in case of polyclonal antibody, we simply have to introduce the antigen and then we will collect the blood, and that blood, from that blood will separate the separate the antibodies which are polyclonal. So different batches vary in composition. So batch to batch there will be difference in composition. Whereas once the hybridoma is made, the production is uniform. Same type of monoclonal antibody you can get. So batch to batch difference is there in case of polyclonal antibody, and there is no such difference is there in case of monoclonal antibody. So we can say that in polyclonal antibody we have a heterogeneous population, whereas in monoclonal antibody there is a homogeneous population. Because it is in polyclonal antibody there is a mixture of antibodies having target against specific epitopes in the same antigen, whereas in case of monoclonal antibody it can target only one type of epitope in the antigen. So in case of polyclonal antibody, since there is a heterogeneous population, there can be high cross reactivity for similar antigens. In addition to the target antigen, it can also bind to similar antigens in case of polyclonal antibody, but no such thing is there in case of monoclonal antibody. So there is low cross reactivity in case of monoclonal antibody. And as we discussed, there is batch to batch variability in case of polyclonal antibody. So there will be low reproducibility, whereas there is high reproducibility in case of monoclonal antibodies. Then you can see this monoclonal antibody with one color, whereas polyclonal antibody of different colors, a mixture of antibodies. So the polyclonal antibody is a mixture of antibody, and each antibody can recognize different epitopes in the same antigen, whereas the monoclonal antibody recognizes single epitope in the antigen. So based on this we can have different points, and you can see here suppose this is the antigen, and this antigen has different epitopes. This is one epitope, this is another epitope, this is another epitope. And polyclonal antibodies, they can bind to different epitopes in the antigen. Whereas if there is a monoclonal antibody, then it will bind to either of them, only one antigen, only one epitope in the antigen. So you can see in another figure. Suppose a polyclonal antibody, you can see here this is a mixture of antibodies, and each antibody is binding to specific epitope in the same antigen. This is the antigen. In the same antigen, different antibodies present in the polyclonal antibody, they are binding to specific epitopes, respective epitopes. Whereas monoclonal antibody is having a single type of antibody which has affinity for the one epitope in the antigen. This is monoclonal. So polyclonal antibody is a mixture of antibodies, a mixed population of antibodies, and monoclonal antibody is a single antibody species. Okay, and polyclonal antibody contain more than one type of neutralizing antibody, so a mixture of antibodies, whereas contain single antibody species in case of monoclonal antibody. And polyclonal antibody can bind to a variety of epitopes, so we can say it is broad spectrum, whereas monoclonal antibodies they bind to single specific epitope. Okay, so you can see here the monoclonal antibody react with a specific epitope on a given antigen. Suppose this is the specific epitope to which the monoclonal antibody binds, whereas polyclonal antibody reacts with various epitopes, maybe this, maybe this, maybe this, maybe this. Okay. Then for polyclonal antibody, we can use different animals like say mouse, rabbit, goat. So many host species options are there in case of polyclonal antibody, whereas few host species options are there in case of monoclonal antibody. And polyclonal antibodies is an in vivo technique, and monoclonal antibody is an in vitro production technique. In the laboratory we produce. So polyclonal antibodies are used in general research applications, antivenom preparations, diagnostics, cancer treatments that can target various tumor cells because of their polyclonal nature. And if we need targeted therapy, so we use monoclonal antibodies for therapeutic purpose. And if we see the stability, polyclonal antibodies are highly stable to pH and buffer changes, whereas monoclonal antibodies are more sensitive to pH and buffer conditions. And multiple clones of B lymphocytes or B cell lineage, and in monoclonal it is single B cell lineage or single clone of B lymphocytes. So this is all about the difference between polyclonal antibodies and monoclonal antibodies. Thank you.